The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 69 of the eLife podcast. I'm Chris Smith from The Naked Scientist. This month, evidence that the brain is potentially crammed full of nerve-producing stem cells, bonobos that have cultural preferences, and the insects that follow my leader. How and why do they do it? But first, there's an old saying in medicine that if you live to 80, then 80% of you will get prostate cancer. Assuming you're a man, of course. But that doesn't mean that 80% of men are going to die of prostate cancer. Some will, but the majority will die of something else completely different. In other words, they don't need aggressive, potentially quality-of-life-changing prostate cancer treatment. And the problem is that when we find prostate cancer in a person, at the moment we don't know who does and who doesn't need that intervention. But that might be about to change. Graham Pockley and Georgina Cosma have used a machine learning algorithm to identify a set of markers on natural killer cells present in the bloodstream that can much more accurately detect who really has got prostate cancer and the likely aggressiveness of their disease.
Prostate cancer affects around 45,000 men each year in the UK, and around 11 or 12,000 men die of the disease. One of the big problems in trying to manage patients with prostate cancer is having a very clear understanding of whether they have the disease in the first place, because the current tests don't work very well. And if they do have the disease, how sort of serious is that disease? Do they need treatment, or can they just be left and watched? So the key problem we were trying to solve is to improve the diagnosis of prostate cancer with using a simple sort of blood test rather than having to take tissue out of patients and looking at it under the microscope.
We do have some blood tests you can use to diagnose prostate cancer, or at least monitor it. So what's wrong with that?
Yes, the most common test is something called prostate-specific antigen, PSA test. So this is a blood test that measures the levels of a certain molecule in the blood. The problem with the prostate-specific, the PSA test, is the fact that in some cases the level is normal in men who have prostate cancer. In other cases it can be raised in men who don't have prostate cancer. So there's a lot of false positives and false negatives, and that causes a problem because it doesn't allow that particular test to be used for screening a large number of men because of the false results it gives.
And what have you done instead to try to work out a way around that problem?
The key thing is we still wanted to go with a blood test because that's obviously the easiest thing to do. So we decided to focus on the patient's immune system. As well as protecting people against infection, the immune system protects people against cancer, and the immune system can recognize the presence of cancer. So we thought that if we took a picture of the immune system in the blood, can that be used to tell whether a man has got prostate cancer, and if he does have prostate cancer, how serious it is?
Georgina, how did you actually go about doing this?
So for this study, we collected and examined the natural killer cells of 72 patients. Amongst these patients, 31 of them had prostate cancer and 41 were healthy patients. So these biological data were then used to produce computer models that can detect the presence of prostate cancer and its severity.
For the test, what we did is we took blood samples and we took immune cells from those patients, and we looked at how they appeared by putting them through an analysis. And so we could identify a whole range of different sort of characteristics or features of these white blood cells, these natural killer cells. And then we wanted to assess whether there are any differences in the profile of those features or those characteristics between men who did have prostate cancer and men who didn't.
And that's where you come in, Georgina.
Yeah, so we initially had a set of 32 biomarkers, and the aim was to find a subset of biomarkers that can be used to predict presence and severity of prostate cancer. We ended up using a technique, which is a computational optimization approach, to identify the set of biomarkers that would make a good fingerprint for predicting prostate cancer. The fingerprint that we found was then used to build a machine learning model.
In essence, then, you know that you've got these features that you can look for that are there, but you don't know which combination are going to be the strongest and most powerful predictors of who's really got disease or not. So having identified the ones you think are going to be the right choices, that's where you then move into building a model so that you can then take this forward and test it against more data effectively to see how good it is, presumably.
Yes, precisely. So after selecting the best candidate set of biomarkers, we developed machine learning based tools. This resulted in a prediction model that was 12.5% more accurate than the PSA test that's used in clinical practice in detecting prostate cancer. We also developed a second detection tool, which was 99% accurate in predicting the risk of disease in patients with prostate cancer.
So, Graham, basically you now have a test which is potentially a lot more accurate than just PSA in isolation.
Yes, that's right. I think generally speaking, the PSA test is only accurate in about 3 out of 10 cases. And so there's lots of men who have a PSA test who are told that they have prostate cancer when they don't. The other issue is that in the vast majority of cases, men die with prostate cancer rather than of it. So it's not, in the majority of cases, a life-limiting disease, but people are then sort of labelled as having prostate cancer and have to live with that for the rest of their lives. What this test allows us to do is sort of identify the presence of prostate cancer in the first case, but arguably more importantly, is we can categorise that, or we can say whether that disease is something to worry about or something not to worry about. And that allows the clinicians to focus on treating the more aggressive disease.
And Georgina, presumably you have road tested this by taking people with an unknown diagnosis and then got the predictions from your model and then followed up with a gold standard, perhaps a biopsy or something from those people to know that you're actually producing accurate predictions.
Yes, we also carried out separate tests using a set of records which were not used during the train and test process. So it's like a mini clinical trial, basically, where we took the finished machine learning model and we inputted about, say, 10 test records and then looked at the output of the model and compared that to their gold standard, which was the biopsy results that we had.
And critically, people are often in a quandary when they're given a diagnosis of prostate cancer as to whether or not they should seek active intervention or a more watch and wait type strategy. Does it help them to make that decision?
Yeah, absolutely. I think that's the fundamental finding of the project. It allows that decision to be made far more accurately. The approach would reduce the need for about 70% of biopsies, which is quite an unpleasant experience. 1 in 20 men who undergo that procedure get some form of infections. So what it would really allow the clinical team to do is to tell a man, you have a very low grade prostate cancer, but it's absolutely nothing to worry about. There's no need to treat it. Or say, you have prostate cancer, it's something we're worried about, and we do need to take it further and then further investigations. But it would remove unnecessary sort of investigations in a large proportion of men. And at the moment, that's just not possible.
Isn't that great news? Graham Pockley from Nottingham Trent University and Georgina Cosma. She's at Loughborough University. In the late 1990s, scientists turned one of the central dogmas of neuroscience on its head. They showed that even in aged adults, new nerve cells are still being born in certain brain areas. Previously, we thought that in higher animals like us, neurogenesis stopped shortly after birth. Now, scientists have made an even more extraordinary finding, that astrocytes, the non-neuronal supporting cells in the brain, can respond to injuries by transforming themselves into cell types resembling neural stem cells and then going on to produce new neurons. Now, it's early days, but it could have huge therapeutic potential. Jens Magnussen.
The brain is very bad at replacing dead neurons in response to injuries. We had found previously that a very abundant type of support cell in the brain, which are called astrocytes, can activate a neuron-producing capacity in response to certain injuries in mice. But not all astrocytes appeared to have this ability, and we wanted to study the mechanism by which some astrocytes produce neurons, because we think that this might in the long term enable us to recruit astrocytes as a sort of reservoir for new neurons that could be used therapeutically.
Traditionally, we think of astrocytes as these supporting cells, but if you injure the brain, you tend to find that they grow a lot and they produce almost like a scar. So that seems to be a little bit contradictory to what you're saying about them now being able to produce nerve cells instead. So are they different sorts of astrocytes that are doing this then?
We think that astrocytes have different programs, so to say, that they can turn on. It's true, after an injury, astrocytes can produce a scar. We found that after a stroke injury, astrocytes can also generate new neurons. And we wanted to understand the molecular program that creates this response.
How did you do the study then? Talk us through what the method was, and then we can perhaps unpick what those results are telling you.
So we used a method called single-cell RNA sequencing. So that's a way to read the entire gene expression profile of individual cells. Now this is informative because cell types and cell states can be identified by the combinations of genes that they express. So we first triggered neuron production by some astrocytes in the mouse brain, and then we looked at their gene expression profiles as they initiated neuron production in mice. We found two things that are particularly interesting. The first thing was that as astrocytes initiated this neuron-producing ability, they first underwent changes that made them extremely similar to neural stem cells. So there are neural stem cells in some parts of the brain that continuously make new neurons, and our astrocytes became very similar to them. The second thing that we found was that even though only a minority of astrocytes went on to generate neurons, all astrocytes appeared to initiate this neuron-producing program, but then halted their development halfway. And we found that we could inject a type of growth-stimulating protein into the brains of these mice, and this helped some of the halted astrocytes to resume their neuron production.
Does this therefore have a therapeutic potential in the sense that you could go into a sector of the brain which is subject to some kind of trauma, some injury, where there's perhaps been some selective vulnerability among a group of neurons, where there's been loss, and stimulate the local astrocyte population to differentiate back into and repopulate that missing nerve compartment?
We hope that in the long term this is exactly what we're going to be able to do. Now we're far from being there now, but if astrocytes can be viewed as dormant neural stem cells, we think this is really exciting because the capacity of the brain to replace dead neurons is extremely low. And if the brain turns out to be full of dormant neural stem cells, then if we can figure out how to unleash their potential, then that might represent a potential way for improving brain repair after injury and disease.
What about the phenotype of the nerve cells that you produce in this way? Are they just generic neurons, or do you end up with neurons that resemble the native population that correspond to that part of the brain where those astrocytes were when you started to stimulate them in this way?
We don't find evidence that the astrocytes produce neurons that are of the same subtypes of the region that they're in. The astrocytes tend to generate interneurons that are not necessarily abundant in the tissue where they're generated. So we think that in order for this to be useful therapeutically in the future, other bioengineering approaches are going to be necessary in order to steer the astrocytes towards certain types of neurons that are needed.
Do you know what the survivability, though, of those newborn nerve cells is? Because when people have studied the generation of new neurons in brains, because that does go on in the adult brain, doesn't it? There are certain areas where neural stem cells continue to give rise to neurons throughout life. It's not a given that the new daughter nerve cells survive. So what proportion of the astrocyte-derived nerve cells survive, and how long for?
It's low, less than 1%. We don't know if this is a shortcoming of this neurogenic process or if this is a built-in mechanism.
And do you know if you can shift the survivability? Are there common routes by which these cells try to die that it may be possible to arrest?
Other groups have found that survivability can be increased by treating the brain with certain chemicals. So I think it's possible in principle.
Jens Magnusson, he's at Stanford University. This is the eLife podcast. We're looking at some of the leading papers published in the journal this month. I'm Chris Smith from The Naked Scientist. Still to come, the first comprehensive review of cholesterol levels in the Caribbean and Latin America. Now, if you were to charter a plane from the capital of the Democratic Republic of Congo, fly for some 4 hours, and then spend 6 hours on a motorbike, and finally travel for a few more miles on foot, you'd come across a field site where there is a unique phenomenon taking place. Two groups of bonobos that have distinct cultural preferences. Eva Hickinbotham heard what they're up to from Lyran Samuni.
As humans, we are this crazy, special, unique species. We are all over the world. We are capable of cooperative behaviors that have no parallel in the animal kingdom. And one of the things that is considered to be uniquely human is our great capacity for culture and cultural differences, which are group-specific behaviors that are acquired through learning. And I think one of the questions is, where in the history, in the evolution of our species, did we develop the tendency for cultural behavior or cultural differences, traditions? When we study the evolution of humans, one way is through fossils. We go to excavation and we find fossils that tells us a little bit about the history of our species. But when we want to look at specific behaviors, it's very tricky with fossils because behavior does not fossilize. And in that sense, we turn to some of our relatives, primates, and especially bonobos and chimpanzees, who are our closest living relatives. This paper looks at what we call diversity of behaviors, and especially one that is emerging between two groups of bonobos that live in the same environment. Each group is specialized on the hunt of different prey. One group hunts antelopes, a small antelope called the duiker, and the other one hunts a gliding rodent. The striking thing is that despite the fact that they live just in the same environment, they hang out in the same places, they still show this group identity in terms of what they feed on.
Is that surprising for animals of the same species that are in the same environment to have that difference in behavior?
I think it's very unique and special, yes, because we can imagine that a lot of how behaviors form and come about is in animals reacting to their environment. If the environment is a certain type of environment, then different behaviors can emerge. But here we have the exact same environment and still almost two different solutions to the same environment.
Antelope are just such different animals from the smaller rodents that the other group hunts. It seems like such a big difference in strategy. Where do you think that comes from between these two groups?
That's a great question, and it's a bit of a mystery. One idea that we had is that meat is a high quality food, but it's very rare and it's hard to attain. So hunting frequencies are quite low, and when they're managing to catch the animals, they're super excited, really hugging each other, vocalizing, and a lot of excitement. One idea that we have of how could this happen, that two groups that live in the same area have such different techniques, is in a way to avoid competition between them. So if we can imagine that there is this high quality food that is hard to access and it's very rare, it might be advantageous for each group to specialize on a different prey, so to avoid competition or to reduce the competition.
What might this tell us about the evolution of culture?
We know by now that cumulative culture, which is knowledge that is transferred from generation to generation, and the culture is shaped through knowledge that is passed on. I think this is something that we don't have great evidence that non-human animals have that. However, the basic capacities of culture, which are two different groups, no differences in their environment, but still showing some diversity of behaviors that is evolved as a function of learning, I think this shows that on the prerequisites of culture as it's seen in humans today. So I think especially the fact that both bonobos and chimpanzees show these abilities tells us a lot about how our ancestors were. Another very important reason why we want to study those animals is in terms of conservation. There is knowledge in the world that is in nature and in wildlife, and we can look at it almost as a library full of books that no one has ever read. And through research, we are able to get a glimpse at some of those books. And I think with the biodiversity crisis, it's almost like imagining that this library will be burnt and this knowledge will be forever gone. So I think in some ways, studying those animals, we are trying to preserve some of this knowledge and at the same time conserve these incredible animals.
A very poignant note from Lyran Samuni, particularly at a time when we're being told that the world is now in something of a biodiversity crisis. Medicine is full of examples where we use data collected on one group of people and then we apply it generally across populations, sometimes incorrectly. Some blood pressure lowering medications, for example, we now know work much better in certain groups of people than others, probably for genetic reasons. Blood cholesterol, or more accurately, blood lipid levels, is another such example. And working in the Caribbean and Latin America, that's what Rodrigo Carrillo has been looking at.
So we wanted to know what was the most common lipid problem in Latin America and the Caribbean. There are plenty of large-scale studies in Europe, US, Asia as well, but in low and middle-income countries like Latin America and the Caribbean, and also in Africa, these sort of quote-unquote well-known questions are still unanswered.
And obviously this is important in terms of a health priority, because if we want to manage disease and anticipate disease risk and therefore plan ahead appropriately, we need to know these sorts of numbers rather than just assume that what's true in North America or Europe applies also to other countries as well.
Absolutely. For these kind of things, we're perhaps still trying to answer the most basic questions, like how many are we, how many are there, where are they? There were some small studies in some countries, but looking at the region as a whole, we still need to answer many questions.
Did the raw material exist for you to study, or did you have to literally embark on this from the get-go, physically drawing blood and looking at people's cholesterol levels to find this out?
In this work, we rely on published data. So we did a systematic review, which in simple terms is looking through every single journal article and scientific paper and extract information when they measure lipids or any sort of cholesterol, to have information from as many countries as possible.
So when you amalgamate all of these research studies, how many subjects, how many people do you end up with to consider?
I think it was about 150,000.
So big numbers, these. And when you extract the data, are there any obvious trends?
Low HDL is not very good for your health, and we found that was the most common trait.
What about the other risk factors like triglycerides and also like LDL, the bad cholesterol that also furs up arteries? What do they show?
For LDL, we also find quite large estimates, quite a large prevalence, though it was smaller than for HDL. For triglycerides, we also find large numbers, and it was interesting to see that triglycerides was a little bit higher in what we call Andean Latin America, which is perhaps Peru, Ecuador, and Bolivia. And it could relate with the sort of diet they have in these countries, which is perhaps most based on carbohydrates.
To what extent do you think what you're seeing here is just the environment in which people live and perhaps their lifestyle. In other words, I know if for instance, you drink too much alcohol, you depress your HDL good cholesterol. And to what extent is this relevant to a person's ethnicity? Because obviously these areas have also seen a lot of immigration over the years, and those people could quite possibly have brought disease risk with them, couldn't they? So how do you dissect out, if you can, those effects?
Yeah, it's difficult to do that with this work, though I think you are right. And lifestyle will have a lot to do with that. But not only lifestyles, but also opportunities to have a better lifestyle. For example, physical activity and also diet. What sort of diet you have access to? Unfortunately, like you said, you can differentiate migration between external and internal migration. And sometimes internal migration coming from rural areas to urban areas, these people do not usually have like the highest standards of living. Of course, these have implications for the diet they can afford.
What are your take-home messages then from what you've discovered? Depressed HDL, some high levels of LDLs, and some high levels of triglycerides as well, and some possible associations with diet. What are your take-home messages, and how should this inform our understanding of heart disease risk in these geographies going forward?
The take-home message is that LDL is important, and many, if not all, clinical guidelines target this with pharmacological treatment. And there is lots of evidence of how lipid-lowering medications work and do great things for your health. But these medications do not usually work for HDL. So the take-home message, I think, is that we have to work together from a practitioner perspective, prescribing medicines when these are required, but also from a public health perspective, in which we should give people the opportunity to embrace a healthy lifestyle with access to physical activity, with access to adequate diet, and to include both in clinical guidelines as well as in policies and strategies. Over the last year, there's been a lot of evidence and a lot of work in obesity and underweight, in blood pressure and diabetes, but lipids and cholesterol overall have not been very well paid attention to.
Rodrigo Carrillo, he works with Imperial College London and also in Lima, Peru. Ants and termites show a behaviour called a tandem run. It's where two of the insects venture out, one leading, one following. If you just glimpse them doing this, you conclude that it was the same behaviour carried out for the same reason. But actually, ants do it multiple times in their lives to teach other nestmates the way to new locations, while termites do it just once after mating to set up a new nest. And for the termites, it's a case of follow my leader. But for the ants, it is different. The leader determines the direction, but the follower sets the pace, so she's got time to learn the way. To discover this, Gabriella Valentini made video recordings to watch the animals running round in petri dishes and then extracted the information about which insect was calling the shots using image tracking software.
This behavior is actually widespread in many insects like ants and termites. They use a very similar communication mechanism. So we have the follower touching the rear of the leader to communicate the presence, and the leader instead emitting a volatile pheromone that can be perceived by the follower. We also know that in ants, the leader is making sure that the follower is learning a new path through the environment. While in termite, they are just navigating an unknown environment. And so the function of the tandem run in termite is simply to maintain a cohesion.
So how did you actually do the study, and what emerged as you did this?
The way we approach this question has been by looking at how leader and follower share information with each other. We performed several experiments where we track leader and followers over time. And what we do has been to look at how the trajectory of the leader and that of the follower are caused by each other by looking at how different type of information flows between leader and follower.
Can you just describe the experiments you actually did?
We did experiment with ants and termites. The setup is different between the two species. Ants are looking for a nest. We are actually starting a new emigration and having them explore an environment and look for a new home. And once they found this new home, they will start recruiting to there using tandem runs. And this is the part of the experiment we were interested in. So we repeat several of these experiments, making this colony emigrate several times. And for each migration, we then recorded the tandem runs and the trajectory of leader and followers. The situation is a bit different with termites. This is because they are not recruiting to a known position in the environment, but they use this behavior only after mating. So we had to collect unmated termites, allow them to mate, and after that, we put them in a petri dish where there was no nest they could find. We recorded the entire experiment using a video camera placed on top of this arena. And from the video recording, then we were able to use a tracking software to extract the trajectory of leader and followers.
And once you'd pieced together all these videos and watched all these ants and termites running, what did you learn?
There are at least two types of information that are being exchanged between leader and follower. One is information about the direction of motion, whether to turn left or right. And another important information is instead about the pace of the run, when to stop and when to resume motion. Although ants and termites can use the same communication mechanism, what we discover is that in the case of termites, the leader is controlling both the direction of motion and the speed. And this all makes sense because we know they are searching at random, and so the input of the follower to that behavior is likely to be of less importance. The interesting discovery was for the case of ants, where we saw that the leader is deciding about the direction of motion, whether to turn left or whether to turn right, while the follower is actually controlling the speed of the run, when to stop and when to resume. And this was a very important finding because it provided support for an hypothesis behind the tandem run behavior of ants, that is that the follower is acquiring information from the environment in order to guide their future journey through the environment. So in this case, we have that the leader is traveling through the environment, choosing the direction of motion in order to show different parts of the environment to the follower.
And why do they therefore need to slow down? What's the benefit of the changed rate from the follower?
We haven't proved yet, but our main assumption, our main hypothesis is that the follower is looking around the environment, looking for landmarks that she can memorize and use as an information to guide her future journey. While they are moving, they have a relatively higher speed, and the follower is very much focused on not to lose the leader. But from time to time, she takes a break. And during this break, the follower detaches from the leader and walks around a few centimeters apart from the leader, usually making a sort of loop before coming back to the leader, touching her gasser, and restarting the journey. So these longer pauses are actually a mechanism that provide the follower with the time to acquire visual information from the environment. And only when the follower has enough confidence in that information, she will then prompt to resume the journey.
Gabriele Valentini, he's at Arizona State University. And that's it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientists. Previous editions, the references and also full-text transcripts for these programmes, as well as details on how you can subscribe to this podcast, are at nakedscientist.com/eLife. And incidentally, The Naked Scientists also publish a weekly science programme that covers the latest leading science news stories. And you can find that at nakedscientist.com/podcast. I'll be back, meanwhile, next month with another look inside eLife. Until then, though, from me, Chris Smith, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.